Well, hello, everyone. Welcome to the April Lunchtime Lecture. I'm Dorothy Guerrero, Vice President of Communications at the Texas Exes and Editor-in-Chief of the Alcalde. Before we get started, we want to invite you to join Texas Athletics, the Texas Exes, and Longhorns in your community at a Texas Fight Tour stop near you. You can hear from Texas Longhorns head football coach Steve Sarkisian and athletic director Chris Del Conte about the future of the Texas Longhorns athletics programs. You can bask in burnt orange spirit and more. So if you want to learn more and register now for our stops in San Antonio, Dallas, Fort Worth, and Houston, go to texasexes.org/tour. And now on to today's lecture, which is called Knowing Who's Boss: How Social Status Impacts the Brain and Health. Our professor today is Dr. James Curley, an associate professor in the Department of Psychology at UT's College of Liberal Arts. Dr. Curley received his BA in Human Sciences at the University of Oxford in 1999 and went on to earn his PhD in zoology from the University of Cambridge in 2003. At UT Austin, his lab focuses on the neurobiological basis of social behavior in groups, as well as the long-term plastic changes in the brain and peripheral physiology that occur as a consequence of social status. He and his colleagues are also interested in developing novel methods for the study of social hierarchies and networks. Please welcome Dr. Curley.
Okay, thank you, Dorothy, for that introduction, and it's a pleasure to be with you all today. I'm looking forward to discussing some of my lab's work. I've been at Austin and in Texas since 2017. Prior to that, I was at Columbia University, and as Dorothy mentioned, I'm British originally, but it's great to be in Texas now and at UT. So my lab is primarily interested in social dominance and social hierarchies. And this is a form of social organization that exists throughout the animal kingdom, including humans. And essentially, these hierarchies form whenever animals compete. And why do they compete? It's usually over access to resources. Those resources may be mates, it could be territory, it could be other things. So animals are constantly in conflict. They need to determine who's dominant and who's subordinate. And they will fight for this access. But the issue with fighting is that it can be very, very dangerous and very costly. You can injure yourself. It's energetically very expensive. So you don't want to be continually fighting. And so what animals tend to do is that once they've determined who is dominant and subordinate, they form this relationship where they may test each other from time to time, but they essentially, one of the individuals will yield and will show a defensive behavior and say, you know, I'm subordinate. And when animals are in groups, large groups, then there's a rank order that exists such that you have animals who are rank 1, alpha individuals. They dominate everybody else. And then you have animals who are rank 2, and they dominate everybody except the alpha individual, and so on and so forth. And throughout the animal kingdom, we find that these hierarchies are highly linear in that you can almost always rank animals 1, 2, 3, 4, all the way down to however many animals are in a group. The classic example was first scientifically described 100 years ago, and that's in chickens, which is like the middle image here, the idea of a pecking order. And I think that that's kind of very intuitive to most people, that chickens have this pecking order. But you know, even in crayfish or in moose, in many birds, we see these hierarchies. So it reduces conflict overall, and animals need to kind of engage in this system so that they're not continually fighting. Okay, now in the lab, it's hard to, you know, have moose in the lab, or it's not that hard to have crayfish, but it's hard to have large groups of them. But we study typically the mouse. And so I'm going to describe how we study the mouse in the lab, and so that you understand when I show you some of our data, how we collect it. Now, typically, wild mice live in burrows. This is kind of a schematic of a burrow system. They dig underground and they create these little chambers. They forage above ground, but they spend most of their sleeping time underneath. And so they're a species that has this kind of social organization. But how do we create this in the lab? Now, at UT, what we do is we have these large vivaria. This is like a metal structure that has a platform level in the middle. I'll use my mouse. If you can see my mouse, it's this middle section. Now underneath we have nest boxes that have tubes that connect. So an individual mouse can run around this whole system. It has kind of underground nest boxes. There's food and water at the top. And that little block kind of top left is about the size of one mouse. So we house groups of males or groups of females, up to 30 animals in setups such as this. And just give you a little video. This is in slow motion, so they're not slow moving mice. This is just slow motion. What you can see is that there's two animals on the left fighting here. One of them is trying to kind of nip and bite the back and the tail end of that one mouse. And that mouse is kind of showing its front, its nape. This is a subordinate posture saying, you know, I submit, don't hit me anymore. And you notice we have these little blue colorings on the markings. This is so that we can individually identify each animal. And what we do is that we house them in these vivaria for 2, 3, or 4 weeks. And we record the behavior that they do. We do this in various different ways. We do it through video monitoring. We do it through live behavioral observation. And we do it through, we also have some tags that we can put under the skin where we can track their movements. Once we track their behavior, what we do is we get kind of data that look like this. So I'll orient you to the matrix on the left first. What we have in the rows and the columns are the individual animals, and this is 12 animals. And on row 1, we see the alpha, in this case male. This is a male from a male group. This alpha male, rank 1, if you look across, the first number you see is 100. And that means that over the 3-week period that it was housed with these other 11 animals, it attacked the second ranked animal 100 times and won 100 times. And a win is if you chase or if you bite that animal. Whereas the animal who was ranked number 2 in the second row, it never beat the first animal. So it never attacked that first animal. So this is 100 wins for animal 1, 0 wins for animal 2. Whereas if you look at, for instance, number 5 against number 6, it beat animal 6 16 times, whereas 6 only beat 5 12 times. So this is a much closer competition. Once we collect all of this data, we can actually say, well, who's the most likely winner or loser? And that's the matrix on the right, where we give a 1 for the winner. And the degree of redness on the right indicates the strength of that relationship. And as you can see from the order of the matrix, nearly all of these 1s are in this top right corner of this matrix, which indicates that we have a really highly linear social hierarchy, where animals can be rank ordered 1, 2, 3, 4, 5, 6, and so on. There's always a few little changes, even in the classic chicken pecking order. Even when you get down to rank 7 and 8, sometimes there are a couple of reversals, but this is a remarkable hierarchy that we can observe in the laboratory. And we've seen this in male groups, we've seen this in female groups. So this is just to introduce you to the kind of hierarchy. I think if you have any questions, please put them in chat as I go through the talk. What I'm going to do is kind of come through 2 or 3 different themes that we study about hierarchies and things that I find interesting, and hopefully you'll find them interesting too. So question 1 is, how do animals know who's dominant? How do they signal to each other who is the dominant individual? And this is not a mouse that you're looking, this is a gelada baboon. And I think one of the first things you'd look at with this baboon is the mouth. You have these huge teeth that look very threatening. And other geladas would look at that and also think it's threatening. But what they're actually mostly focused on is the red chest patch. They're looking at this redness and how red that chest patch is. The degree of redness correlates to how dominant an individual is. The most dominant individual has the darkest red on that chest patch. So this is just one example. This is from a recent paper that I published where we characterize different ways of signaling. So in the bottom right here, I have the gelada, and on the most bottom right is a mandrill. The way mandrills signal their dominance is through the degree of blue and redness in the face. Other visual systems include fish and lizards. So the anole lizard on the left has this black eye spot, and the cichlid fish has a black eye spot as well, an eye bar actually, it's a black bar across its eye, as well as the coloration, the yellow and bright colors indicate that that's a dominant individual. Other species use vocalizations, so you can see the red deer and the chacma baboon in the top right are vocalizing, just shouting and making these calls to say they're dominant. And other species use smell. So in the top left, we have a ring-tailed lemur. They have scent glands where they scent against objects or trees or whatever it might be in their environment to indicate I'm dominant. And then we have the mice that we study. They also use smell, which I'm going to discuss in a bit more detail very shortly. So there's lots of different ways of signaling dominance, but they all really have one thing in common, and that's this, which they need to be energetically costly to produce. So what we're looking at here are red deer. So I've already mentioned that they vocalize, but another way that they display their dominance is through these antlers, huge antlers. Now, individuals with the largest antlers are typically the most dominant individuals. These are very energetically costly to produce. You wouldn't just want to produce large antlers, on the off chance you might be the most dominant individual. You should use that energy for other purposes. So these signals have to be energetically costly, because otherwise it's just pointless producing them. But also it would be too easy to fake if they were not. If it was, for instance, the eye spot in a lizard, if that was easy to turn on, then everybody could do it. And so this is why they're costly. So let's turn to mice. How do they signal their dominance? They actually have such a protein or a family of proteins that they put into their urine. And these are called major urinary proteins. They're very, very large proteins. What mice do is similar to kind of your dogs. When if you ever take your dog for a walk, you may notice it urinates against the same lamppost every single time, over and over again. Mice do the same thing, but not lampposts. They're usually urinating on territory boundaries, which might be, in this picture on the left, is like a branch or a plank of wood. And that kind of bump that mouse is sniffing is actually just protein from the urine that's just laid on top of each other over time and creates this bump. And this is a mouse marking its boundary territory. And the proteins are called major urinary proteins. There's one of them, which is number 20. It's called Darcin, after Mr. Darcy from Pride and Prejudice. And this protein attracts females. So if a male urinates this protein, females find that urine and they're looking for the male. They find it attractive. This same one and also another one, number 3, signal dominance. So if a male comes up to another male and smells this protein, it knows that that animal is a dominant, because dominants are the ones that produce this. And one reason that dominants produce it and subordinates don't is it's so energetically expensive to produce. If you're a subordinate, it's a waste of energy to even try producing these proteins. Now, the other key thing about these proteins is that they bind small molecules. These are volatile odors, organic compounds that evaporate into the air very rapidly. So by binding to these proteins, they stay longer, which means that once an animal has marked its territory with urine, it doesn't all just evaporate immediately. It's slow releasing because of these proteins. Now, in our lab, we've been interested to find out, well, what are the chemicals that are released in urine to signal dominance? And we've teamed up with an engineer at UT, Pavel Mitzel, who has this amazing machine. It's one of the most sensitive machines of its kind, where you can pump air through it, and millisecond by millisecond, it will give you details of thousands of chemicals that are in the air. Now, Pavel uses this a lot to measure pollution down in Beaumont, Texas, Corpus Christi, but I managed to convince him that it would be a good idea to study mouse urine as well. So he allowed us to do this. And we have this setup that looks like this. I'm going to show you a video. This one, unfortunately, looks like it's shot from the moon. But in the top right, we have a mouse. It's a dominant mouse. And on the left, we're seeing a couple of graphs of 2 chemicals that I'm interested in. On the right, several other thousands of chemicals being recorded. But what you notice on this spike is that our animal here just urinated. And then all of a sudden, we got this huge uptick. And this is kind of, as I said, millisecond by millisecond analysis of the chemicals that are in the air that this mouse has emitted. And these 2 chemicals are called DHB and SBT, not particularly interesting. But we've identified that these are 2 volatile odors. These are compounds that are airborne, that are only produced, or, well, I should say, produced in huge quantities by dominant individuals. So this is a graph showing the spikes of these chemicals over time. And we can see there's basically 4 different peaks. The blue is one chemical and the orange is the other one. And what these correspond to are when the alpha male was put into the chamber, then taken out, that's the first huge peak. When a subordinate male was put in and then taken out, that's that tiny second peak. The tiny third peak is a different subordinate male being put in and then taken out. The huge 4th peak is a different alpha male being put in and taken out. And then we have these 2 quiet flat periods either side of the peaks. And this is where we had dominant and subordinate females in the chamber. So these chemicals are produced, these are male chemicals. They signal dominance to other males. And we have now found over 1,500 different chemicals that are emitted by mice. We found chemicals associated with male dominance and chemicals associated with female dominance. So it's kind of, we feel like we're really getting to the bottom of this question is, how do these animals signal their dominant status? So the next big question we have is, well, if these are the chemicals that they're producing and we know that they can signal to each other, how does the brain actually encode these signals? And I was fortunate enough to, this year, I received an NIH grant where we're able to study this question. How does the brain encode this? Now, I'd like to kind of introduce this by talking about this one study in humans. Now, in humans, we're a hierarchical species too. We perceive social status and who's more dominant or less dominant than ourselves. And so these researchers, this is not my work, this is work of others, ask the question, how do humans encode social status? So to do this, you can see in the bottom left, C, we have these different pictures. We have 7 faces of same generic, young adult male. Well, 7 different young adult males, but they all look quite similar. And then we have 7 pictures of galaxies. And the participants of the study were told this is the rank order of these subjects. And they were tested over and again. So they might see face 3 and face 5. And they were told, and they had to learn that 3 was dominant to 5, 2 was dominant to 6. They did the same for the galaxies. So that red galaxy at 4 was dominant to the green and yellow one at 6. And so they learned the rank order of the galaxies. They also learned the rank order of the humans. And then in the imaging phase, when they were put into an imaging machine, they were asked to recall who was dominant to who in different tests. And we see the results on the right. And I'm just going to highlight up here this finding that they had, which was that this one brain region, the amygdala, was particularly activated when humans were observing dominant individuals. When they were identifying humans in rank 1 and 2, the amygdala. Now, the amygdala is a bunch of different nuclei. It's a brain region that is particularly involved in processing emotional information, particularly social emotional information. If you're very fearful, if you're very anxious, particularly in social context, this is a region that's highly activated. So this is very interesting that this brain region seems to have a role in regulating dominance. Now, in the rodent, we were asking the question, how is smell processed and is the amygdala relevant? Smell related to dominance. Now, I need to tell you one thing about the rodent brain that's different to the human brain. Whereas in the human brain, our capacity of smell, we can only really smell airborne odors. So we call these volatile. So like if you smell burning toast, that's coming to you through the air, that smell. We don't do what dogs or horses or other animals do, which is pump up fluids, pheromones into our olfactory system. We can't process pheromones, but the rodent can. So this is a top-down view of a rodent brain. And there's 2 pathways. There's this one pathway for detecting these airborne odors, volatiles, and then there's this other pathway for detecting pheromones, these like the major urinary proteins, the big dense chemicals that you need to literally pump up into your olfactory system. And that's why animals sniff each other in the way that they do. Now, the key thing about both of these pathways is that they converge on the amygdala. If you see my mouse, it's basically this. It doesn't really matter, but it's this oval. You have one on each side of the brain. And so this is where these odors converge. So we tested this by doing this particular study. Now, what we do here is that we expose mice to different urine. That urine is either from an alpha individual or a subordinate individual. And there's another wrinkle, which is that they're either individuals that they're familiar with or individuals that they never met before. And we tested animals who were somewhat dominant themselves in purple and or somewhat subordinate themselves in orange. And what you can see with the 4 box plots all on the left is that they're much higher than the 4 on the right in this one region, medial amygdala. And what we're measuring here is something called CFOS reactivity. Now, CFOS is a protein that's produced by the brain when that brain region is activated. This protein gets produced. It's kind of our imaging method. And we can see here that when these animals are exposed to alpha urine, dominant male urine, this brain region, this particular part of the amygdala, the medial amygdala, it was just highly activated. So this is really good evidence that this brain region in mice, similar to in humans, is activated when processing dominant cues, but it's not really that active when processing subordinate cues. So we're now following this up with methodologies where we actually dissect this region and we look at the neurons and we try to identify which particular neurons are being activated. There's more to this story. There's a nice brain region I think is interesting, which is called the periaqueductal gray. If you stimulate this brain region, it will make an animal freeze and show a fearful response. So this is the main role of this brain region. And what we can see here is that when animals were exposed to dominant urine from an animal they'd never met before, so an unfamiliar alpha urine, both animals who were quite dominant and subordinate showed higher activity of this brain region. So it seems that sometimes animals use this brain region to kind of invoke a fearful response. You should be scared of a dominant male you've never met before. So we're now trying to track down more details about the circuitry behind how the brain is processing such cues. Okay, so these are how animals produce these signals. But if you're going to urinate as a mouse to signal your status, there's one thing you have to do to be able to do that, and that's drink a lot. You also need to eat a lot because you need to produce the proteins. And what we're looking at here on the y-axis are 12 males living in a social group. And each little blue tick represents when they were eating or drinking. The first 12 hours is during the dark period for the mice. Mice are nocturnal, they're more active during the dark period. And the second, the right-hand side of the graph, is the light period, kind of our daytime, but then nighttime. And as you can see, the rank 1 animal has the most blue ticks, and it goes throughout the 24-hour period. Dominant animals just have to eat and drink constantly to maintain their social rank. In order to urinate this frequently and produce these proteins, they just constantly eat and drink. Another method we use is, you can see on the left, we have these cages. They have these kind of antennas and wires connected to them. Like you might have with your cat, like my cats, they have little micro tags under them to ID them. We can put those into mice, and as they run through little setups like this, we can track when they move through different cages and different tubes, and we can track their movements constantly. By doing that, we've shown that dominant individuals are basically just whizzing around the whole, they're constantly patrolling. They're observing everything, looking, where is everyone? Is there any intruders? Constantly moving around. And dominant males spend far more energy patrolling than subordinate individuals. So how do they cope with all of this energy that they have to use? Well, one method we looked at this was to look at the liver, which is a part of the body that is very involved in energy production. And we dissect the livers out of them. We extract RNA from the liver, and then we sequence the RNA for every single gene in the entire genome and look at how much of each gene is expressed. And the most striking thing we found was that alpha males in their liver, the genes that are involved in fatty acid oxidation and lipolysis, which is the breakdown of fats, so burning fats for energy and breaking fats down, they were hugely upregulated in alpha male individuals. That's that purple top graph, the purple dots. Whereas subordinates were actually trying to conserve energy. The yellow dots there represent fatty acid biosynthesis. They're trying to produce fatty acids. They're trying to conserve energy. And there's this complete shift in dominant and subordinate individuals in how they're dealing with energy. Dominants are just going for it, and subordinates are trying to conserve. These are just some individual genes just to get a little bit more into the biology. Basically, how fatty acid oxidation works is that mitochondria, which are the powerhouse of cells in the liver, there's this particular pathway called carnitine that it brings in fatty acids into the mitochondria. They then burn them and break them down to produce energy. All of the genes involved in this pathway, alphas, are just, upregulating. They're just going for it, basically. Okay, so a little summary of what's going on with these dynamics is that being a dominant male, you have to drink more, you have to eat more, you're patrolling. I didn't mention, but in the blood, they have higher testosterone and growth hormone, 2 hormones. These stimulate this production of these proteins that they use for signaling in the liver. This is where they're produced. The liver is also burning fatty acids and breaking down fats. Then these proteins get excreted in the urine. Urine is also controlled by the brain. I didn't mention that today, but the brain is also changing to control their urination. So basically, dominant males have a pretty miserable life where they're constantly just trying to stay dominant. And I like to think of it like this, which is from Henry IV. Uneasy lies the head that wears a crown. It's very, very costly and precarious to be at the top of a hierarchy. And I've given you my example from my mouse work, but we see this in primates, in other species. It's it's difficult being at the top. Okay, so it's difficult being at the top, but it's also not a bed of roses being at the bottom of a hierarchy. So what are the long-term consequences of being socially subordinate? This is some data from humans. So it's actually social economic status is one of the strongest predictors of mortality in humans. So the graph on the left is looking at survival curves of the top 95th percentile for income and the bottom 5th percentile for income. It's a huge difference in survivability if you're in the lowest status versus the highest status. And this is because status and SES is strongly predictive of so many different diseases all listed there. Many, particularly heart disease, but also various other ones as well. Now, this is data from the US, but this is not about wealth per se, it's about status. This is a pre-industrial society that is studied by ethnographers in Bolivia, the Chi-Manay, and it's been shown in this population that although everybody is pretty similar in terms of wealth, there's still strong status differences between individuals in terms of a hierarchy in the society. And these health differences are still apparent, with people at the bottom of the social hierarchy suffering from worse blood pressure, cardiovascular risk, and so on and so forth. So being at the bottom of a hierarchy is worse in terms of survivability and health. We can see this across all different species. This is just a summary slide from many different primates and sheep and goats, rodents. Being at the top of a hierarchy, you live longer on average. There's some differences, but on average, if you're at the top, you do live longer. Now, why is this? Well, one strong hypothesis that has a lot of support is because if you're at the bottom of a hierarchy, you suffer from damage to tissue that is caused by high levels of stress hormones that are constantly circulating. This is a graph that looks at savanna baboons. And what we can see here is that the lowest ranked individuals, 13, 14, 12, have higher levels of stress hormones, which are called glucocorticoids. This is from fecal matter, but it's also true from blood. They have higher levels than more dominant individuals, with the one exception of the most dominant individual actually in this study has the highest stress hormones. But generally speaking, being at the bottom of a hierarchy is a risk factor for high levels of these stress hormones. Here's just a little bit of data from my own lab. On the right, we see in females hierarchies, the subordinates have higher levels of stress hormone, corticosterone, than the dominants. On the left, it's split into two panels. This is because our male hierarchies, we have some male hierarchies where the alpha male is definitely dominant, but he's a little bit more relaxed and allows other animals to fight each other. We have other hierarchies where he's despotic and he refuses to let other individuals engage in any fights with each other. In those hierarchies, we see our subordinates with the most divergent stress profile. They're much more stressed than if they're with a more relaxed alpha male. So we see this too. Subordinates have high levels of stress hormones. What are the long-term consequences of this? Well, in primates, which have been well studied in the wild and in the lab, it's kind of mirrored the human findings. Subordinate macaque monkeys, they have higher levels of atherosclerosis. This is like thickening of the arteries. They're more at risk from viral infections. They keep colds for longer, and that's incredibly dangerous for primates in the wild. They don't get rid of these infections. They also show reduced wound healing. If they do get injured, and subordinates do get injured more than dominants, they just don't recover from injuries as quickly. So higher ranking individuals are much faster to recover. So there seems to be some severe long-term outcomes for being subordinate. At a molecular level, some colleagues and like Jenny Tung, who used to be at Duke, is now in Germany, has done some lovely work looking at the gene expression profiles of these monkeys. And she has shown from blood cells that if you're a subordinate individual, you have higher levels of genes that are involved in inflammation. And you have lower levels of genes that are involved in antiviral defense. And this might explain why animals who are subordinate are not doing well at overcoming viruses, and why they're suffering other diseases, because they're constantly inflamed. Okay, so this is some data from my lab kind of getting at this question. It's a tricky graph. I tried to think of other ways to present it, but let me see if I can explain. So what we're using here is a method called flow cytometry. We take blood samples from dominant and subordinate individuals, and we put them through this process where we sort the cells. We say, what type of blood cells do they have? There's lots of different types of blood cells. And we count up how many they have of different types. Now, in the orange, these are the adaptive immunity cells, like B cells and T cells. These help in things like viral defense. They're the things that produce antibodies. And we see that dominant males have higher proportions of those. Those are those red lines to the right-hand side. They have higher proportions of those cells. Subordinates, on the other hand, don't have as many of those cells, but they have more of what we call innate immunity. These are things like the neutrophils, you can see here with red. These are the things that whenever you have like a bacteria infecting you, they go to the site of the injury and they rush there and try and attend to it. So subordinates are investing in this innate right now immunity, whereas dominants are investing in the adaptive viral defense for later. Now, at a genetic level, we've looked at peripheral tissue. This is what primate research can't do, but we can do in the mouse, is that we can look at tissues like the liver, like the spleen, and see which genes are activated. And for instance, in subordinates, we find that they have higher genes like ILB interleukin 1b, which is an inflammatory marker, and they have higher expression of genes that also inhibit the production of lymphocytes. These are these white blood cells like B cells and T cells. So subordinates are really getting a bad deal here. They're inflamed, and they're not able to produce as many B cells and T cells. So this is actually an area of research that we're trying to get some funding for, because it seems like we have a great model for being able to study the health outcomes of social status, and not just look at the blood, but actually look at tissues themselves. So a little mini summary before I spend kind of 10 minutes on the last topic. So what I've shown you so far is that animals are able to shift status. They can become dominant or subordinate. I'll get into how quickly this occurs later, but it can be on very short time scales. Sometimes it takes longer to emerge into dominant and subordinate positions, but it has huge consequences for behavior and physiology. This goes beyond just aggression. Dominant individuals, they need to change behaviors like eating, drinking, signaling to others that they are dominant. And this requires massive energy production and changes to liver physiology, for instance. Whereas subordinates try and conserve energy, and this might have some short-term benefit, but it's also associated with long-term negative health outcomes. You're constantly exposed to stress hormones that leads to tissue damage, inflammation, and the inability to cope with viruses, and also this increased cardiovascular risk. Okay, so kind of like the next little thing I'd like to talk to is, well, it seems like it's really bad to be at the bottom of a hierarchy. It's probably good to be at the top of a hierarchy, and it certainly is good for animals. And the reason why it's good to be at the top of a hierarchy for animals is that they are the individuals who get most of the mating opportunities and are able to pass their genes down. But how do you get to be at the top of a social hierarchy? So there's many different ways. I'm really just going to focus on one in detail, but let me just briefly outline the others. So, like the country that I come from originally, the one way that many animals do this is inheritance. So like our royal family is passed, being monarch is passed down generation to generation. In particularly female primates like macaque monkeys, but also this amazing species, the spotted hyena, which is a very female-based society where the females have the strongest dominance relationships. You become dominant, you inherit the rank of your mother. So if your mother is a highly ranked individual, when you become of age, you take her place and get that rank. If she was at the bottom, unfortunately, you become a bottom ranked individual. So this is one way that you can become to the top of a not a very fair way, but it's one way. The other is prior attributes. And these prior attributes are things like fighting ability or boldness or body size. It's that simple. Like gorillas, if you're the biggest, meanest gorilla in the troop, you have the largest sagittal crest, which is this crest in the top of the skull that has the musculature of the jaw all associated with it. You are likely to become the most dominant gorilla. The bottom left are elephant seals. If you want to have the most matings of females as an elephant seal, you need to be the biggest elephant seal on the beach. That's the way elephant seals become dominant. Similar things in goldfinch, the birds there, or crayfish, as you see there. So that's another way. But not to be overlooked at all is that in many animal societies, you may also say in human societies, it's just random luck who becomes dominant or subordinate. And so let me explain a little bit about how it can just be random luck. And that's through what in our field we call experiential factors. And the most classic of these are winner and loser effects. So a winner effect is if you just won a contest or a fight, the probability that you win the next fight, everything else being equal, opponent quality, everything else being equal, you get this boost from winning the previous contest. Whereas if you lost that previous contest, you get this loss. And I'm actually doing some statistical work now on online chess. I play a lot of online chess very badly. But you may have noticed that if you've ever engaged in something like that, if you win a game, you sometimes seem to win 2 or 3 in a row. Whereas if you lose, then you might lose 2 or 3 in a row. It's similar to the hot streak in basketball 3-point shooting. If you hit one, it seems like you hit the next 2 or 3. Whereas if you miss one, it's hard to get going again. These are winner-loser effects. And we see this across the animal kingdom. I have a number of papers where I've statistically analyzed data from fighting behavior and shown that it exists in chickens, in fish. And now there's evidence that loser effects are stronger than winner effects. And that being, if you lose, you maybe really should pay attention because, you know, it's bad to lose and it might be dangerous to keep losing. Whereas winner effects, they exist, but they're a bit more subtle. I don't have too much time to go into this, but this has been very nicely studied in fish, the best evidence. These are the species of fish. For instance, in the Amazon molly, you can do these experiments where you take the same size fish and you pair them either with really small ones or really big ones. The ones who are with really small ones, they win lots of fights. The ones who are with big ones lose lots of fights, and then you start pairing them together. And you find the ones who had the experiences of winning, they keep winning, but those who had the experiences of losing, they keep losing when they're facing equal opponents. And in guppies, a collaborator of mine, Ivan Chase from Stony Brook, has done really nice work where he showed that guppies form hierarchies. And then he took them all apart, gave them a few months where they kind of forget each other, don't know each other, put them back together, and showed that they form completely different hierarchies. The rank order of the animals was just jumbled up. And this is because one of the reasons as to why animals became at the top or bottom was just this luck, winner-loser effects. Okay, so the last thing I want to really talk about for 5 minutes or so is social skills. And these are fundamentally important in dominance hierarchies, regardless of whether you're at the top or whether you're at the bottom. Now, one strand of my research is that we do a lot of work looking at early life experiences and how it predicts later behavior. And just like in children, in rodents, this is rough and tumble play on the left. These rats, these are juveniles. They're not fighting, they're play fighting. You see this in cats and dogs as well. They're learning to fight, but they're doing it without risking injury to each other. And it's been shown that rats that do more of this play fighting with each other, they become better at real fighting when they're adults. It reminds me, this is a bit of a British specific quote, but it was said that the Battle of Waterloo was won on the playing fields of Eton by the Duke of Wellington, Eton being a school in England, basically saying all the games that they played, the rugby and the contact games and strategic games they played in the schoolyard, helped them learn how to be great army generals when the time came to be army generals. Okay, so how do we go about looking at how animals use social skill in social hierarchies? Well, there's a few different ways. Coalitions, I'm not going to spend too long on that, and how they monitor relationships, and also this idea of taking advantage of when social opportunities occur. So in brief, because I'm running out of time, several animals use coalitions. Mice don't use coalitions, but lions need to collaborate with other male lions to take over prides. Chimpanzees, there's famous examples of how the second and third ranked individual will team up together to overthrow the top individual, like you're seeing in this top left. Even in very large brained birds, such as crows and ravens, they form coalitions with each other. And obviously in humans, my favorite example, I like ancient Rome history, the first triumvirate. Caesar, who had the least power at the time, cooperated with Pompey, who was an ex-army general, and Crassus, the richest man in Rome, to take power. And then once he formed the coalition, he obviously broke the coalition. But monkeys and other animals, so I'm going to use some monkey examples here, they observe social relationships. So what's going on in this study is that the researchers played playbacks of animals shouting at each other. And they didn't really pay attention when it was a subordinate losing a fight to a dominant. But when they switched the sounds around, so it sounded like the subordinate was beating the dominant, all of a sudden, all the monkeys in the troop suddenly turned around and looked at the speakers, like what just happened? And this is evidence that animals are constantly paying attention to social interactions. Another example is transitive inference. What this is that if I told you that A beats B and B beats C, you wouldn't need to see that A beats C. That would be something that you would likely infer because we're able to use transitive inference. I don't have time to go through the experiment because of time, but we've seen this across the animal kingdom. This is some nice work done by Hans Hoffman, who's actually at the biology department here, showed that cichlid fish can do this. If they watch other fish paired up, fighting with each other, they see A beats B, B beats C, C beats, they can then work out that A is likely to beat E and B is likely to beat D and so on and so forth. And even children, I won't go through this again, but children as young as 10 months old are able to do this. We can show that if they see a relationship that looks like it's wrong and that a lower ranked individual has suddenly beaten a top ranked individual, they start looking at the screen for longer and going, what's going on? Showing that children are primed to recognize hierarchical relationships. But we do see this this notion of attention hierarchy. And the key thing that's going on here is that subordinate individuals need to monitor dominant individuals in a group. They need to know what is the dominant doing because it matters to them. They don't want to get on the wrong side of a dominant individual. This is a nice study in Rhesus monkeys that shows that in the lab, what they have to do is that Rhesus monkeys, they get grape juice for a reward. So when they're shown photos of gray squares, just a boring gray square, they're not interested. They don't want to see the gray square. They would rather have the juice reward. If they get shown a male face and that male is low status, they're not interested in that either. They still want the grape juice. If they get shown a female sexual swelling of a female, they want to see that. They're not interested in the grape juice. They give up the grape juice. But interestingly, from kind of my perspective, is if they're shown a dominant male's face, they don't want the grape juice. They want to look at the dominant male's face. So they don't care about low status animals. They care about high status individuals. We see this in children. This is the study, but I won't describe it. But children also spend most of their time, these are preschool children in preschool, looking at dominant individuals. We see it this is data from my lab in my mice. What we have here are blue ticks represent when the alpha male is being aggressive. An orange tick shows you when other males are being aggressive. And what you can see here is that they're offset from each other. The subordinate males in my groups they're not aggressive when the alpha is being aggressive. They avoid him, they watch him, they monitor, and then they wait until he's inactive, like eating or drinking or sleeping, then they're aggressive. Because this is a social strategy. Okay, so the last thing I want to tell you about is social opportunity. And this is when individuals recognize that a power vacuum has arrived in a hierarchy, particularly at the top of a hierarchy. But power vacuums can exist in the middle of hierarchies too. And what subdominant individuals need to do is that they need to pay attention as to when their opportunity exists. So to give you another ancient Rome example, the Emperor Vespasian in AD 69, after Nero committed suicide, there was the year of the 4 emperors, where 3 different emperors tried to become emperor, but they didn't have the support. They didn't manage to hold on to power. And then eventually Vespasian was able to usurp all of the others and become the emperor. So he rose up the hierarchy when this power vacuum emerged. Now we see this in primates. Although in rhesus, they generally wait to become dominant. It's called laddering up. They sometimes will try and just overtake if a dominant individual kind of falls. And what we see here is the same monkey in these 3 photos. In the top left is in his prime. The second one on the top is when he lost a fight and was starting to realize that he was no longer going to be dominant. And then underneath is him looking really sad. He lost his position. The graph on the right, these lines are different dominance ratings. You can see this line at the top. He was for a long time the dominant individual, and then all of a sudden just crashed all the way to the bottom of the hierarchy. Other individuals recognized his fall from grace, and they jumped in to become dominant in his place. And we see this this happened over months in the primate, but in the cichlid fish that also form hierarchies, it can happen in seconds. Within 30 seconds of a dominant going, a subordinate can become brightly colored, get the eye bar, increase testes size, have these hormonal changes to get to the top of a hierarchy. Now, we've also started, for the last few years, looked at this in mice. And what you see here is 3 days worth of data looking at 12 different mice. And that red line at the very top, the dark red, is the dominance rating of the alpha male. It's a very high rating over 3,000. It means he's very, very dominant. And you can see there's a dashed line underneath it. That's the beta male. He's the second ranked male. When we take the alpha male out of the group, so he's just gone forever, what happens is that the beta male becomes the dominant, rises up, realizes within 10 minutes, starts attacking everybody else, I'm now the alpha male, and another animal rises to become beta, second, ready to be alpha. If we remove that alpha male, then the next one rises up, and there's a second rank who becomes beta, and so on and so forth, all the way until we end up with 2 individuals. Do we get this situation where animals will just rise when the power vacuum emerges? And the reason we're doing this in mouse is because we can ask what in the brain changes when this happens. And again, we use the CFOS mapping technique to look at the brain activity. And what we've shown in the orange bars here, when animals are ascending, they're socially ascending, they have hugely, like just dramatically increased activity in infralimbic and prelimbic cortices. These are the parts of the brain right at the front of the brain, the frontal cortex. It's like we have a much larger frontal cortex, obviously, than a mouse, but it's the front of the brain. And this is dramatic. And what we've done in more recent work is that we've looked at the genes that are also changing. So that this helps us to hone in on which neurons are changing when an animal sees that it's their chance to grab power, which neurons become activated. So our goal now is to try and manipulate these neurons to see if we can make animals go up or go down a hierarchy just by stimulating these neurons. So just to summarize this kind of second mini section, there's different ways to get to the top of a hierarchy. Simplest being just be big and good at fighting. That works in some species. Experiential effects are surprisingly strong across all species, and that's just luck. Whether you just managed to win by, luck that first fight, or you lost that first fight, that can really set you on different trajectories. And obviously, if you happen to be in a society where inheritance is powerful, then that's a very easy way to get to the top or the bottom of a hierarchy. But the other thing to note is that when you live in a social hierarchy, you need to use social skills. You need to form coalitions in species where this exists. You need to constantly monitor other social relationships to check who's more dominant to who in that group. But you also need to monitor when there may be a social opportunity for you to jump into that power vacuum. And as I've just shown, we've started to identify those brain regions that appear to be monitoring such opportunities. So with that, I would like to just acknowledge largely these 4 PhD students who have worked with me. They've all been fantastic students. I'd like to thank all my funding that I've had for my research, and thank you for the opportunity to talk to you, and I hope that it was of some interest.
Thank you, Dr. Curley. This is really fascinating. We have some questions from the group I'd like to put out there. So the first one is, have you examined cultural differences in how humans display dominance, for example, East versus Western cultures?
I have not studied that, but it has been studied. I'm not an expert on it, but I can give you some general kind of themes. So body posture is one way that dominance is used across cultures. So obviously the classic, broad-shouldered upright posture has been seen to be a dominance pose. Another one is eye gaze, so averting eye gaze, and there's many societies, in many societies, avoiding eye gaze is used by subordinates to not intimidate dominant individuals, but particularly in several Asian cultures, avoiding eye gaze is seen as a much bigger deal than in some Western cultures. In fact, certain faces are seen as more dominant. So the facial asymmetry, or particular in males, I don't know why I'm looking at my face, but like a masculine, very broad-jawed face is seen as dominant. And there's some data that shows that you can throw up on a screen these images, and the participant won't even recognize that they've seen a face. But you can ask them, was that dominant or subordinate? And they'll just think they're guessing, but they guess far greater than chance, which goes to show that the human brain is processing these images like subconsciously almost. So I guess what I'm saying is that there's many different cues that humans use to do with facial, to do with voice, to do with eye gaze. And there are some cultural differences in how they're used. I'm not currently an expert on lots of those, but I am writing a popular book on social dominance. So I need to become an expert on that question because I want to include it in my book.
Great. Okay, we have another one. What prevents subordinates' bodies from producing equal levels as the alpha in order to challenge the alpha's dominance?
Right, that's a really good question. So there's two ways of answering that. Number one, sometimes physiologically they just cannot do it. If you're a young immature monkey, you just aren't able to produce the cues like the red chest patch, or you just can't do it. You don't have enough physiological capability. But sometimes there are be it monkeys or the mice, they are able to do it. Now, the main way is that dominance. If you're a dominant individual, you check. So if I'm a subordinate mouse and I produce these chemicals, I decide I'm going to produce them. Well, the dominant will attack you. It will notice that you've produced them. It will start attacking you. So the way in which they get shut off is actually the social suppression of being attacked. It's actually, well, I won't go too much into it. There's some very interesting questions about the neurobiology, but generally speaking, the social stress and social suppression of receiving aggression shuts down these pathways. And it would be a bad idea to turn them on because you will be checked by the dominant.
Okay, I love this next question. Have you watched Survivor and been able to predict the winners from the socialization?
No, no, but I do know about it, but no. I once got asked about Hunger Games, and that was another TV show that I didn't know. My wife knows much more about TV shows than me. But I have seen the premise. Now, the one thing I would say about, I don't know that much about it. The one thing about that, which is interesting to me, is that there's coalitions, right? So individuals have to form alliances, and then it's about forming alliances and breaking alliances. And as I mentioned, we see that in humans, we see that in many primate species. There's a few other, like lions, when males and these birds, ravens. But that's where it's very, very tricky to predict when alliances form or break off. In fact, colleague of mine in the anthropology department here, Aaron Sandel, studies chimpanzees in Uganda, and he and his studying these chimpanzees in the wild, they found that the group just split in two. And so some alliances formed between some chimpanzees and others, and then they had a big falling out and they split. And the question of trying to predict how alliances form and how they break is actually really, really hard. So I guess if I was watching that, my frame of interest would be on why certain individuals, what social skills they're using, because you have to be liked to form alliances, but you also have to be able to know when it's your turn to, turn on your colleagues, I guess.
All right. Here's another one. What about research on type A and type B personalities? Aren't type A people more likely to be dominant, but also prone to heart disease?
I don't know about their proneness to disease. I could look that up. That's very interesting. There is definitely personality types in terms of dominance. There's a wrinkle to human dominance, which I didn't get into, but I can briefly mention now, which is that in animals, what we talk about dominance, we're largely talking about physical competition. So that's, I beat you because I'm physically stronger. You do see that in preschool children, like the work I briefly mentioned of Patricia Hawley, who's at Texas Tech, because preschoolers will push each other or pull each other. In humans, dominance is harder to discern because adult humans don't often go around pushing or shoving each other. But there's another stream to dominance, which is called prestige. And prestige is dominance by reputation, or how well thought of you are, how well liked. Now, getting back to the type A and type B personality part of the question, is that when we look at middle childhood into adolescence, the children who are perceived to be dominant tend to be those type A, pushy, more pushy individuals who are have this dominant personality. But they then don't end up being the ones who are liked. And so the ones who end up having dominance by prestige tend not to be that type of individual. So it's a different type of dominance. And in terms of the disease, what we're really looking at, there's many reasons why individuals may have certain diseases, but the human data in terms of status is about kind of social stress that induces this hormonal profile. So that's why we see that association. It's because of the association with constant social stress hormones.
All right, I think we have time for about one more.
Mm-hmm.
So in humans, are there biological factors that predefine our behaviors, at least to some degree, that are either difficult or even impossible to change or overcome?
I'm going to go with a firm no on that. I don't think there are any. I think that, so big picture here, as you get older, as you develop, things obviously become harder to change. and things are much easier when you're young or when earlier. But the brain is, it's plastic, particularly until your mid-20s, but there's always ways of changing. It's quite a big question. There's a few ways I could answer it, but one way I think the take-home message I would like people to have is that the brain is very plastic, and these biological correlates can be shifted. In fact, the things I've talked about, dominant, so like this, and subordinates like this, well, you could be a subordinate who becomes dominant, and then your brain will change. The other thing I'd like to point out is that people have tried in humans to identify biological correlates of dominance. So there are certain genes. One example is monoamine oxidase A. It's an enzyme that breaks down neurotransmitters like dopamine, serotonin. Now, people have associated that gene with serial killers and people that show violent conduct disorder. There was a particular famous family where many of that family were all, incarcerated for serious violent crimes, and they all had this one mutation in this gene. So then people got carried away and said, well, it looks like this gene is related to aggression. But the thing is that if you look at different environments that people grew up in, you could have that mutation and be completely, show none of that behavioral profile. And so it's very much about how these biological predispositions are shaped by the social environment or the physical environment that you're in that ends up with how your behavior and your brain and your physiology. So I would say none of this is ever causative, and there's always ways in which things can overturn or be modified over time.
Well, Dr. Curley, thank you so much for your time. This has been
a pleasure. Thank you.
A lot. Thank you all.
Thanks.